All right, this video is sort of a compilation of a few different sections to sort of put a lot of ideas together. So this is going to we're going to talk about how molecules pass through the plasma membrane. So if you remember, the largest component of the plasma membrane are the phospholipids. So we have a phospholipid bilayer. And this is a selective membrane, which means not everything is going to pass through it. So in that portion of our plasma membrane, where we have phospholipids, so I'm just drawing a quick small section, there are certain things that will use the phospholipid bilayer in order to pass through. And certain types of passage are only going to be allowed here. So in the phospholipid bilayer, this is where diffusion occurs. Diffusion. So we talked about several different ways that things can pass through a plasma membrane. Diffusion is one of those. Diffusion will use the phospholipid bilayer of that plasma membrane. There are a few characteristics of the molecules that will go through that. So if you're going to use diffusion through this, you're going to have characteristics that will allow you to do this. One of these is you need to be small. And the second one is you need to be nonpolar. Or a lot of times I will say non-charged. Because to be nonpolar means you do not have a charge. So some examples of this are things like gases. There are a lot of gases that will diffuse through this phospholipid bilayer. Things like nitrogen gas, oxygen gas, carbon dioxide gas. Now, we don't want to get nitrogen into our system. That can cause us problems, but we definitely need to get oxygen into our cells in order for us to do cellular respiration, or at least aerobic cellular respiration. So, O2, CO2, N2, these are all nonpolar molecules. None of these will dissolve in water, something we talked about. So they break down there because you notice that they're small. They do contain highly electronegative elements. But if you have oxygen on the outside of your cell, it's able to fit through those spaces between the phospholipids. Or carbon dioxide, when we want to get it out of our cells, it can also fit through those parts. So being small allows you to fit in between here. But the other thing that's important here is these are nonpolar. Remember, nonpolar do not interact with polar. This is why oil sits on top of water. It doesn't want to interact. It wants to be here and it wants the water here. So as these carbon dioxide molecules get close to this polar head, because this has no charge, it doesn't interact with that polar head. So it doesn't get stuck to it like something polar would. Remember, if you're polar and polar, you're going to be like magnets. This has no magnet, but this side has a charge. So these phospholipids have charges. My carbon dioxide is neutral. My oxygen is neutral. So not only are they small and they're able to fit between these, but because nonpolar do not interact with polar, they don't get stuck on the outside with the polar heads. All right, a second way, or second thing that we'll use our phospholipid bilayer. Sometimes these can be a bit of a tongue twister. Osmosis can occur through the phospholipid bilayer. Remember, osmosis is a special type of diffusion. It involves water molecules going from a high concentration to a low concentration. Now, water breaks the rule a little bit. Water is small, but water is polar. But it is so small that it doesn't get attracted to the polar head. So even though it's polar, it is so small it doesn't interact with the polar head. So remember, so in this case, osmosis also sort of has a special condition. Water is small enough at H2O that it can still sneak between these molecules, and it can go both directions. Water can also come into the cell this way, depending on whether or not it's a hypotonic or a hypertonic situation. Because it is so small that it sort of overrides the fact that even though it's polar, it can still sneak between those phospholipids in the bilayer. And then we have a third way we use this phospholipid bilayer. All of that bulk transport. So a third way that we can move things in and out using the phospholipid bilayer is by bulk transport. And this can go either in or out. So remember, we have exocytosis in order to go out, and we have endocytosis to come in. And that can either be phagocytosis or pinocytosis or receptor mediated endocytosis. And if you recall, this is when we're going to merge our vesicles with that phospholipid bilayer. So I'm just going to make my phospholipid bilayer as two lines. If this is the outside of the cell and I have a substance out here and I want to bring it in, so this is when you will have an invagination into our plasma membrane. So that substance will get really close and it will sink into that plasma membrane. And then as it continues to sink in further, it will become wrapped up into a vesicle, and the plasma membrane will go back to being its normal self. But this is also using the phospholipids. A vesicle is a small compartment. It's a membrane-bound compartment, and so it is made up of phospholipids. Or substances could go the other way. You could have endocytosis. Sorry, that was endo. You could have exocytosis, where a vesicle that has a substance merges with the plasma membrane and then will eject the substance out of the cell. So the three ways that we use to move stuff into the plasma membrane using the phospholipid bilayer are 1, diffusion, 2, osmosis, 3, bulk transport. All of these are using the phospholipid bilayer. So what is the other or the next thing that is most prominent in our membrane? And those are the proteins. Proteins will use some of the other forms of moving stuff in and out. Excuse me. So if you have your phospholipid bilayer, these are going to all be integral proteins. So they're going to span, and then it could either be a transport or a carrier within that. Anything that spans the phospholipid bilayer is an integral protein. Two that will help us move things in and out are the channel and the transport. Because both of these go the entire length, they are both integral proteins in our membrane. So here's our phospholipid bilayer. Here's a protein going the entire distance. Let's make the other half. So there are going to be two major ways that we're going to move things in and out of those proteins. One of these will be facilitated diffusion. Remember, facilitated diffusion is when we use a protein to move stuff in and out. So now instead of moving in between the spaces of the phospholipids, we're going to have things go in and out of this protein. Facilitated diffusion will always take place in channel proteins. So they will have no interaction. This is just like a sewer pipe. If you have a lot of stuff on this side and a little bit on this side, things are going to flow this way. No interaction. If there's a lot of stuff on this side and a little bit on this side, it will flow this direction. Or, if you recall, we can also have active transport. And in this case, we will have interactions or we may not. So these can be either channel or transport proteins that will allow active transport. So both of these have molecules that are similar. If you use either facilitated or active transport, the molecule characteristics are still the same. I think I'm only going to have room spell characters. So you notice with diffusion and with our osmosis, it was about small and uncharged. The opposite is going to be here. We're going to be typically large and charged. And by charged, we mean you can be polar or you can be ions. So the reason that polar molecules and ions have to use proteins is because they get caught up on here. If you have a polar head, it's going to be attracted to a polar molecule, and then all of a sudden you're going to try to get in, and guess what? You get stuck on the polar head, or you're going to try to get out. You get stuck on the polar head. If you get stuck on the polar head, you're not going to be able to move through. So because of the chemistry of this phospholipid and having that polar head out, things that are charged get stuck there. Therefore, they need a neutral space in order to cross. So if you're something like a sodium ion and you want to get in, you would get stuck on a polar head. But if you have this free space of the protein where it's neutral, it's like the DMZ in South Korea, you have a free passageway to get in or out. Or if you're a potassium ion and you're on this side, you would also get stuck to these polar heads. So you move out through the protein. Because you notice these are small. You would think they would be able to squeeze through. Turns out they can't because they're so charged, they can't get through. The other things that will move through this way are large molecules. So glucose is too big to squish through. Plus it's polar, so it gets stuck and it's too big. So larger molecules like glucose, they're also going to use proteins in order to move in and out of our system. And then one last molecule that we have to think about. There's a special type of protein. It's called an aquaporin. And as the name suggests, this is going to be a protein that specifically allows water through. So, water can move in by osmosis, but water can also use facilitated diffusion. So water, if it is on the outside, not only can it go between, water can also use very specialized proteins called aquaporins. Aqua is in the name. That means it's a specialized protein that allows for water to move through proteins through that plasma membrane. So sometimes water will come in between molecules. Sometimes it will come in between proteins. Or if you also remember, you had bulk transport, and pinocytosis is a third way we can get lots of water into a cell. So water, because it's so essential to life, can come in by diffusion, facilitated diffusion, or pinocytosis. But typically if you're moving through proteins, you're either doing facilitated diffusion or active transport. It's for large and charged molecules.